You're fully capable of feeding the cats yourself. Oh, hi. In this video, here are our objectives. We will give an overview of protein synthesis. We will give a brief review of transcription. We will utilize mRNA and an mRNA decoder to simulate translation, not transcription, the next part, translation. And we will complete transcription and translation for a segment of DNA. We will follow that by summarizing transcription and translation. But let's start with some vocabulary so if it's used, you'll understand what we're meaning. mRNA is the messenger RNA. It's a nucleotide that is synthesized during transcription. It uses DNA as a template to create its own code. tRNA, the T stands for transfer, so this is transfer RNA. A nucleotide that complements mRNA and uses its code during translation to assemble the proper amino acids required for a specific protein. Codon, the three-letter code found on mRNA. Anticodon, the three-letter code found on tRNA. They complement each other. So just like DNA matching up with DNA and DNA matching up with mRNA, we've got mRNA and tRNA matching up nicely. And our last term is amino acid, and it is a subunit of a protein. Just an overview of protein synthesis. If you recall, we've talked about the nucleus. What's inside the nucleus is this double helix structure, double-stranded structure known as DNA. It has these nitrogenous bases that complement each other. The A's always go with the T's. The C's always go with the G's. But then we talked about transcription, where the DNA opened up and created a strand of mRNA, where instead of the A's, instead of the A's matching up with T's, the A's now aligned with U's. T's did not exist anymore in mRNA. And now to the bigger deal for this video, and that's translation. Translation happens here at this thing known as the ribosome, where that mRNA is now used as its own template so that this structure here known as tRNA can complement it and bring along with it an amino acid. So if they all bring a different amino acid and they get linked together like a chain, you are now building a protein. I do encourage you to go back to the last video where I showed you this video from YouTube from the DNA Learning Center about transcription. It was very helpful. Just to recap myself, transcription is when that double-stranded DNA opens up and just a little part of it unzips to reveal the nitrogenous bases of the DNA, for which now the RNA will set up its own sequence of nitrogenous bases. But again, remember, whenever there's an A on DNA, there becomes the complement is a U on RNA. It's a big deal. There are no T's. So now our focus is really going to be on what's happening out here in the cytoplasm at that ribosome and so forth. So what happens to the mRNA after it leaves the nucleus? Well, I'm going to go back to that DNA Learning Center, and I'm going to allow them to help us once again.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules.
So those big structures, those green structures that are floating around, it seems like it's aimlessly. Well, they have attached to them an amino acid. Now, it's not aimlessly. They will find a new home.
There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain.
Do you see that? The amino acid that comes with the tRNA is left behind when the tRNA leaves.
Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
It's actually quite remarkable that it does that. So did you witness that the tRNA brought with it an amino acid to the mRNA? And that's how they, well, built the code. So now the big question is, how do we know which amino acid is associated with each of the tRNA molecules? See, in this illustration right here, it's just calling it amino acid, but they have a tool. But let me just throw this at you. Actually, let's just get right to that tool. All right, so this is a codon decoder. By taking all of the 3-letter codes of the codons found on an mRNA strand, you can determine the amino acid sequence used to synthesize proteins. Anticodon decoders also exist, but they're different. They show that code. So let me go back to this. And if you notice, this is the mRNA, but they're chunked by 3s. These are called codons. So these on the tRNA are called anticodons. And just like we've been doing with all the other times with the nitrogenous bases, they complement each other. The U's go with the A's go with the U's, the C's with the G's, and the G's with the C's. Notice, I didn't mention T's at all. That's only in DNA. So, how does this thing get used? We need to look at these letters on the left and top and the ones on the inside. So, how many codons? And we'll understand how to use this by actually applying it. First question has to be, how many codons will be formed by the stretch of DNA that reads TAC, ACC, GCA, TAA, and ATC? And remember, it's in triplets. So if I just go every 3 and draw a line, I'll identify how many codons. So 1, 2, 3, 4, 5 codons. What's this other question? What is the amino acid sequence for this? Well, in order to do that, we first need to do that transcribe. And where's my eraser? There it is. So we actually have to transcribe this. So the A would line up, the T would line up with an A, the A with a U, the C with a G, and I'm just going to go all the way through this. G, C, G, U, A, U, 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 A, and G. Now remember, this is now mRNA, and mRNA, when at the ribosome, are read 3 at a time. So this is AUG. AUG. Which amino acid would be attached to the tRNA if the mRNA was AUG? Well, we look for the AUG here. We have A, we have U, so that's here, AU, AU, AUG. It's right there. So the amino acid would BMET. Now, that's the start for every single one of them. Sometimes they have it listed as start, but that's the first amino acid in all proteins. What's the second? UGG. We go to U. Yes, UGG. UGG. The second amino acid is TRP. The third, CGU. CGU. It's right here. The fourth, AUU, AUU. That's a capital I, then an L and an E. And last, UAG, UAG, and it's stop. What that means is that tRNA molecule actually does not have an amino acid attached to it at all. So there's nothing for the following one to attach. So it actually severs the cord. And so this amino acid sequence is these 4 amino acids right there. Now, you see how that code works. This is based on mRNA, but so is this one. And these code decoders can take many different forms. And to prove it to you, let's just do one example. What about U leads to that amino acid known as PHE. So UUU, and the same thing right here. So they are consistent. They are the same thing. So I've now given you the tools to figure out from mRNA which amino acid sequence we have. So let's just start from the beginning. Check for understanding here. And we'll use this code right here. And using the above code, let's answer the questions. So what type of nucleic acid is this right here? And I need you to provide evidence. Well, this is DNA. And the evidence is we have T's available. There are no T's in RNA. How many nucleotides are present? Let's take a count. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. 18 nucleotides are present. Now from that, how many codons are present or can be generated from the strand? Now, it goes every 3, right? So that's 1. I'll do a zigzag line to represent the next. 2, 3, 4, 5. We have 6 codons. I think that's the last question there. Yep. Let's move on to some more. All right, so here they are. This time I have them color-coded, so they're a little bit easier. Here are the 6. How many anticodons are present or can be generated from this? Well, if we could get 6 codons, we must be able to get 6 anticodons. How many amino acids will be part of this peptide chain? And remember, this last one, as we discussed just earlier, is going to be not an amino acid at all. It's going to be stop. So it's going to be 5 amino acids. Now I'd like you to compose the mRNA that would be transcribed from the given DNA. So take a moment. From this DNA, transcribe it into the mRNA. Pause the video and give it a shot. All right, here it is. Hopefully I didn't make a mistake. What goes with TAC? AUG. The complement for ACC is UGG. The complement for GTA is CAU. The complement for AAC is UUG. The complement for TGG, ACC, and the complement for ACT is UGA. Now, from that, let's use the decoder below and figure out the amino acid sequence. So, the first one, AUG, where are you? AUG, right there. The sequence begins with MET. Peptide bond, UGG, UGG. Uh-oh. Nope, it's a good one. I thought it hit a stop. And that is TRP. CAU. CAU goes with HIS. UUG. UUG right there. Next amino acid would be LEU. Peptide bond bonding them. That's a U, not an O. ACC. ACC is right here. The amino acid is THR. My penmanship is horrible today. And lastly, UGA. UGA is a stop. So we have 1, 2, 3, 4, 5 amino acids in this chain. All right. Now let's compare and contrast. We have transcription on the left, translation on the right. What do they have in common? Well, they're processes in protein synthesis. They utilize A, U, C, and G as their bases. And they need templates. The transcription has DNA as its template, and the translation has the mRNA is the template for the tRNA. What do they have that's different? Well, its product is mRNA. Transcription makes mRNA, while translation, its product is protein. Transcription happens in the nucleus for human cells. Translation occurs in the ribosome. Now, the reason why I mentioned in humans, for all animals, I should say, bacteria, it's a little bit different. That's another subject. And lastly, transcription, DNA makes mRNA. Translation, mRNA makes tRNA, or at least helps tRNA find a location with its amino acid. So it's making a protein. So let me show you these now. This came from past regencies, and it asked what molecule is represented in each picture. Up top, which molecule is this? I hope you said DNA. And I hope you did that for a couple reasons. We have T's present, and it's a double-stranded molecule. What is molecule 2? Well, I hope you said RNA. At this stage, mRNA would have been just fine, but RNA for certain. And molecule 3, I hope you wrote a protein. Let's take a look at this picture. This is also from a past Regents. What process is A? We go from DNA to making mRNA. That one is transcription or translation? Transcription. C comes before L. Transcription. And right down here, process B, we have mRNA combining with many tRNAs with their amino acids, producing a polypeptide. Hope you realize this is our translation. And I think I have one more slide. Yes, just one more. Here it is, starting with DNA, leading to RNA, leading to polypeptides, which are proteins. Sometimes those proteins are enzymes or other cell parts. What is this process called where DNA goes to RNA? Again, transcription. RNA leading to polypeptides, of course, translation. And here's another shoot. It's saying DNA leading to more DNA. Process 1 would be our DNA replication. Horrible spelling. You get the idea. All right, the kid didn't feed the cat. I got to go do that. Have a great day.